So now we'll talk more specifically about hypothesis generation. Now we've kind of talked about how we interact with these programmatically. So let's take an example of explaining a significant overlap between two gene sets. So say we have two gene set libraries, and so a collection of gene sets, and we want to see, are there any interesting associations that we can find between these two gene set libraries? So the first thing we're going to do is look at the two gene sets and see, is there a significant overlap, which we can determine with the Fisher's exact test. And if there is, then let's construct a prompt to try to get GPT-4 to create a hypothesis about why these gene sets might overlap. So we have the gene set terms, which are going to be very applicable to the meaning of the gene set, right? So this is sort of how the gene set is defined in text, very basically. And then we have two maybe abstracts or descriptions of how the gene sets were created. So this, if they were extracted from a paper, then you might want to use the abstract of a paper. If it's a library such as GTEx, you could sort of explain what GTEx is and how those gene sets were actually created. And then we can also look in those textual descriptions to find specific biomedical terms or named entities, such as genes, diseases, or drugs, and we can provide those to the prompt as well. So it might focus on those more specifically. And another use we can take those for is by querying PubMed. So we can query PubMed with those terms to see if they're co-mentioned in literature, to see if this is sort of a relationship that has already been explored extensively. And once we pass this to GPT, we can get some explanation based on those abstracts and based on the terms to some possible shared mechanism between the gene sets. So just some sort of basic notes and a broad overview. The quality of your input and how you're producing that input is going to quite directly impact the quality of your hypothesis. So you really want to have good data to actually do a task like this with. There's many sort of analyses that we can do at the data level to insert into the prompt to improve these models' responses. So we'll talk a little bit more about that. And we can also think about multi-shot prompting in a context like this. So like I said a little earlier in the lecture, asking it to reconsider its answer or think more about it or find flaws in its logic can often help to improve the final result. So we've integrated a hypothesis generation like feature on many of our sites, which I'll sort of go through each one a little bit more specifically now. So Rumagene is a resource that has automatically extracted gene sets from PubMed central publications, specifically from their supplementary material. And this is, a very large collection of gene sets. So this is currently, as of the 10th, so a few days ago, there was 710,000 gene sets extracted from 133,000 supporting tables. And when we were writing the manuscript for this site, we wanted to ask this question of, can we find any unexpected overlaps between gene sets extracted from different publications? So you can imagine crossing 600,000 gene sets is quite an overwhelming amount of sort of relationships to consider, right? So we thought about how can we sort of prioritize some of these gene sets? And since we have abstracts for all these papers, we have some textual description of these gene sets. So we thought, can we quantify a level of surprise that two gene sets are actually overlapping? So what we decided to do is take these abstracts and process them. So this is using some natural language processing techniques such as lemmatization, removing stop words, and then creating term frequency, inverse document frequency vectors, which are basically just vectors representing the amount or the count of words and how important they are in a piece of text. We can then actually calculate the cosine similarity between those vectors, and therefore the similarity of each abstract to each other abstract. So we did this process, and then we looked at sort of the top pairs that we found, and we found some common trends in them. So one was that we were seeing a high percentage of these sticky proteins. So these are proteins that are showing up very commonly in these proteomic assays, and therefore there might not be as functionally relevant, or they might not be really important to look at in this context where we're looking for these surprising relationships. We also looked at okay, are the genes that we're seeing in these top pairs just really highly expressed? Are these being, commonly reoccurring from different omics experiments? And we see in general that there is actually a higher percentage of these top expressed genes. Um, we also looked at the top cited genes. So the top 500 cited genes from GeneRiff, which again sort of keeps track of those gene mentions and publications. That's what we queried with the MyGeneInfo API earlier in the lecture. And we can see that there's actually a lower percentage of these top 500 cited genes, which kind of coincides with what we saw with the expressed genes. So, well, how are we actually getting a hypothesis out of this? So we've made some observations about these top pairs. So let's create some hypotheses about them. Well, one we can look at gene sets that have in their term. So the term here is actually assembled from the column name of the supplementary table from a paper. So it's a little less descriptive, but in some cases, or in many cases, there was a gene in that name, or there was a certain disease in that name. So we're going to look at specifically gene sets that have that gene or disease mention. Um, and we're going to provide GPT with a hypothesis or a prompt that includes the terms. So what PubMed article they're from, um, uh, the term that we identified, so that gene or that disease, um, and then also the abstracts for the two papers. And we're going to say you should draw upon both of these abstracts, the gene and disease terms, in the context of these two gene sets that are highly overlapping, or significantly overlapping, but have very different abstracts. So you can see that that actual part of the prompt is defined at the system level below here in this messages array. So let's look at an example. So these are two of these gene sets that we've extracted. So you can see in this first, up at the top, we have glioma. And we also have gene mention clue here in the second gene set. So those two were identified when we were looking at these gene set terms. And we can also see that if we query PubMed with clue and glioma, that there are zero actual results there. So this association has, as far as we can tell, not been observed. And we can see by reading through the hypothesis here that it generates that the clue gene is involved in mitochondrial function, and that this involvement in the function of mitochondria may be involved in the progression of glioma, or that's what it's going to hypothesize. So obviously, this isn't a sort of confirmed hypothesis or anything like that, but it is a plausible explanation of how this gene and disease may be connected. And that's really what we're getting at, is a starting point for experimentation and a plausible explanation of why these gene sets might be overlapping. So another example of sort of using these Rumagene gene sets is in the context of D2H2, or the Diabetes Data and Hypothesis Hub. So this is another site that we've developed that has hundreds of studies in human and mouse concerning diabetes and other related metabolic disorders. And one of the features that we implemented on this site, because it's very focused on hypothesis generation, is this daily automated hypothesis. And what we do is we take a differential gene expression signature from one of these studies that we have on D2H2, and then we submit it to Rumagene through its API, and we get the top 100 results, and then we rank them by abstract similarity, and then we take the most dissimilar result, and we create a hypothesis with GPT-4. So this looks like something like this on the site. So we have like a hypothesis from April 10th here, and it looks very similar to what we just saw in that example from the Rumagene crossing, except that this first signature is actually now from a specific GSE, where we're comparing two conditions. And you can see it sort of is going to format its response in a similar manner, where it's going to have the hypothesis, it's going to apply that to both the abstracts, and then have some concluding text. And we actually offer a module on the site below this that lets you insert your own gene set and your own description of your gene set to do a similar sort of workflow. So I talked about supplementing uh, the hypothesis or prompt with some data level analysis. So in this case, we could think about giving it terms from certain enricher libraries that are significantly overlapping with our overlapped genes. So what are some functions that these two gene sets might have in common? And we can try to determine that at the data level before giving it to GPT to try to form this hypothesis. So we're still going to use those descriptions and the gene terms, but we're just also going to have some enricher terms that might describe shared biological pathways or otherwise in there as well. So this is something that we've implemented on a couple different sites. One of these is G2SG or the Gene Set to Go site. And one of the features it has is this common fund gene set crossing. So we talked about the common fund before, and we've created various gene sets from different DCCs. And one of the features it has that you can cross them and look for these significant overlaps. So here we can see that we're looking at glycosylated proteins from Glygen, as well as GTEx tissue-specific aging signatures. And we can see that there's this significant overlap between this one glycosylated protein and the gene set involved in blood from comparing a young blood to older blood. And we can see that we've inserted these different enricher terms from Wiki Pathways and from Go, as well as from mouse mammalian phenotypes into our prompt. And then the model is actually going to draw upon those to supplement our hypothesis and to try to identify why those sort of terms might be shared or how that plays in to maybe the shared regulation happening in these two gene sets. So the way it's implemented on this is that you can then actually go hover over that term and see the significance of that overlap with the overlapping genes from those two sets. Another place we've implemented this is for RamaGeo, which is a newer resource that we've developed, which has sort of hundreds of thousands of differential gene expression signatures from geo studies that can be searched at the data level. So with your gene set, as well as at the metadata level with text. But now if you submit a gene set and you have a textual description for it, we can perform a very similar task where we're going to give it to enricher first, the overlapping genes, to find some terms that are significant with those overlapping genes from Wiki Pathways, from Go, so looking for these shared biological processes. And then we're going to insert that into the prompt for the model to draw upon when creating its hypothesis. This is something that you can play around with as well on ramageo.com.